Manager at Nanostring, back for another NanoNews break at the AACR meeting, coming to you live from New Orleans, Louisiana. We just got done with our Spotlight Theater presentation, where our Chief Scientific Officer and Senior VP of R&D, Joe Beechem, presented along with our two customer speakers. We have Arutha Kulasinghe, who is a research scientist at the University of Queensland in Brisbane, Australia. And then we have Hargita Kaplan from Atreca Inc., a biotech in the San Francisco Bay Area. So it's so nice to have you here today. So for those who missed the Spotlight Theater, could you start out perhaps, Arutha, with giving us a little background and summary of what you presented on today?
Yeah, absolutely. So we looked at, my lab really focuses on immuno-oncology, looking at predictive biomarkers of response to therapy. And we do a lot of work in COVID-19 as well, so looking at rapid autopsy samples and really trying to understand where the virus is locating to in those tissues.
Mm-hmm. And I understand you use GeoMx as well as CosMx Spatial Molecular Imager. Like, how do those two data sets interplay?
Yeah, no, great question. So we use the whole transcriptome GeoMx-based approach for the discovery part of it, and then with those findings, and when we want to get granular with the data, we go into the CosMx and then look at the cell type based analysis. So it really connects the two platforms together really well.
Excellent. And then one of the great features of the GeoMx DSP is the ability to do functionally distinct tissue architectures. So what was your strategy behind selecting regions of interest and then segmenting them?
Yeah, great question. So for the GeoMx, we use a masking function. So if we're looking at solid tumors, we gate on cytokeratin, and then we're able to capture the tumor regions. And then we can use CD45 or non-cytokeratin to capture the stroma from those tissues. So we effectively get the tumor and the stroma for those samples.
And what about the COVID-19 samples? How did you profile those?
Yeah, great question. So we used RNA-scope to identify where the virus was in the tissue, and then we honed in on those regions of interest, defined them, and then used the CosMx as another layer of sort of deeper information on different cell types that are being infected by the virus.
Excellent. So Hargita, I understand that you did some T-cell receptor profiling, which is kind of a new addition to the GeoMx digital spatial profiler assays. So could you tell us a little bit about your talk?
Absolutely. Our lab was chosen. I work for Atreca. We're a small biotech in South San Francisco. And we have a novel antibody in a phase 1b clinical trial right now called ATRC-101. And we've been developing the GeoMx DSP so that we can use this on our clinical trial samples. Because of all the optimization and development work that we've been doing, we were invited by Nanostring to participate in your your TCR alpha program.
Mm-hmm.
So what we did was we took some commercially procured samples and a control tissue, and we ran them on the GeoMx DSP using the WTA profile.
Mm-hmm.
And we got some results, and thanks to your wonderful team, we're looking at the analysis, but it looks like we've got some pretty positive outcomes.
Excellent. And I understand that Atreca is sort of like the superstar in terms of running the most number of samples on the GeoMx DSP, so you guys are experts.
At this point, yes. We have worked up both the nCounter panels for protein and RNA for immuno-oncology, and are now working with the RNA WTA panels. And we're really excited to look at the TCR information, and we're more excited to actually be able to use this on our clinical samples, because it's really going to take our exploratory efforts with our clinical tissues to a higher level.
Yeah, I mean, you're going to really be able to understand at a more granular level, like what is the difference between people that respond and don't respond. And also to help discover, biomarkers for, efficacy or new potential targets for additional drug development.
Exactly.
You mentioned in your talk that because you've run so many samples, you've really seen, the reproducibility of the genomic system. So I was wondering if you could comment on that.
So we have run both the protein and the WTA on multiple different solid tumor types: breast, colon, ovarian, melanoma, and lung. And we've noticed that no matter what we run, no matter which ROIs we select, no matter how we segment it, the data, reproducibility data, is amazing. It's exactly, it lines up, it looks beautiful. There's some outliers, but that's expected in everything. But after you normalize it, it's a beautiful straight line.
Oh, that's excellent. We love to hear that. And, it's sort of a continuing tradition here at Nanostring with our, direct digital counting on the nCounter. So I wanted to talk to both of you briefly about spatial biology, right? So it's a big buzzword lately. And, what really excites you? We'll start with you, Arutha, about spatial biology.
Absolutely, great question. I think for us, what's really exciting is the ability to do biomarker discovery work in any disease model. If you have the right cohort, the potential is incredible, and we've shown on multiple studies the potential of spatial biology for that discovery work. And what it potentially means is that you'll identify new targets for certain therapeutics in cancer or potentially even COVID-19. And for us, that's really, really exciting. So every project is, it's novel, it's custom, and it's we're so excited to see the data come out and, find the nuances in there.
Yeah, and I think one of the things too that I wanted to touch on, which you talked about in your talk, is you could have two biopsy samples where if you're doing bulk expression analysis or sequencing, you could get sort of the same expression patterns at that scale. And I think you showed two FFP biopsies where it had the same tumor inflammation signature score, but one is immune excluded and the other is immune infiltrated. And you would lose that granularity if you didn't do spatial profiling.
Yeah, no, absolutely. We see that on multiple projects. When you mash up the tumor and do whole genome or whole exome, the data you're able to extract from that's very limited.
Mm-hmm.
And you really need the imaging with the genomic and proteomic readout to really understand what's going on, especially in immuno-oncology where certain structures within the tissue, such as tertiary lymphoid structures, and being able to really hone in on those and to understand those are really important. So getting that imaging and the other sort of sequential data sets is critical.
Yeah, and exactly. I also wanted to mention, you touched on this in your talk, that tissues, not blood, is where immune cells function, right? So it's great to look at blood samples and PBMCs, but really where the functionality happens and where the immune cells are fighting disease really occurs in the tissue. Yeah. So, people for decades have been looking at imaging, but with, high-plex spatial profiling with GeoMx and CosMx, we can really look at much more markers than we were ever able to before.
Absolutely.
So, Hargita, I wanted to ask you, what do you think the future holds for spatial biology?
Well, I think it's huge. All the lovely work that is going around here, especially by my colleague here and other researchers in the field. Even in the clinical application, that's what we're going to be using it for. The fact that you can actually look at an area on a tissue sample, look to see where there's infiltrating tumor cells and infiltrating immunocells, and actually be able to extract that cellular data mm-hmm. and then map it back to the structure that it came from is huge, and it's going to expand on everybody's view of reevaluating tissue. I totally expect pathologists to be staying in the field for a long, long time. Even with digital pathology, this is definitely another level for pathologists to really understand infiltration of tumor cells and non-tumor cells and immune cells.
And I think this will be really important for extending the work that's been done with cancer immunotherapy. Cancer immunotherapy has worked really well in certain cancers. We have adoptive cell therapy that works really well in hematological cancers. But, getting those immune cells to really infiltrate the tumor is really, I think, a hindrance. And for some people that have an immune excluded tumor to begin with, doing this type of profiling on the tissue level and really understanding that, I think will help improve the efficacy of treatment by leaps and bounds. And at the end of the day, we are all here at this conference because of the patients.
Right.
And I thought it was really great in your talk, Hargita, where you said that you started out in cosmetic chemistry and then moved over to cancer based on your experiences. And I've had my father, my grandfather, my uncle all have prostate cancer. So I think everyone at this meeting has been touched by someone who's been affected by cancer. So thank you so much, both of you. Spotlight Theater presentations were excellent. It was awesome to talk to you. I hope you enjoyed the rest of the meeting and can't wait to see the great things that come out of your two respective labs.
Thank you for inviting us.
Hello everyone, I'm back again, Elizabeth Schneider, Senior Global Marketing Manager here at Nanostring for another NanoNews break coming to you live from AACR in New Orleans. And we just interviewed Arutha Kulasinghe and Hargita Kaplan post Spotlight Theater, but we wanted to dive a little bit more into the project that Hargita did that was part of our TCR Profiling Alpha program with Nanostring. So we have here obviously the lovely Hargita Kaplan from Atreca Inc. She's a research scientist there. And then we have Jason Reeves, who's a data scientist at Nanostring. So I'll start with you, Jason. Could you orient us to what this TCR profiling assay is and how it's used with GeoMx DSP?
Right, so before GeoMx DSP, the only way to really get any information about understanding the diversity and clonality of your sample type was to destroy it.
Mm-hmm.
And so one of the things that GeoMx does exceptionally well, obviously, is to look at the tissue and try to dive it into it physically with probes and understand what genes are being expressed in different locations. We decided that it would be a great idea to extend the fantastic performance of the WTA and the CTA assays so that we could actually start to capture the diversity and clonality of T cells themselves as they are living either inside the tumors or outside the tumors, and think about how they may be influenced by their microenvironment. So the panel that we built is a panel of over 200 probes that specifically target the alpha, beta, gamma, and delta variable and joining change segments. They also include a panel set of 60 different negative controls so that we get exceptionally clean background signal and know exactly which targets are expressed where. The whole idea of this panel is to give you the ability to understand clonality, diversity, and see clonal expansion, contraction, and immune response to the microenvironment physically within your sample.
And what GeoMx assays can it be spiked into?
So this is completely compatible with the Cancer Transcriptome Atlas, the Whole Transcriptome Atlas, and even our small panels can use this as well.
Oh, I didn't know that.
If you have a custom NGS assay, anything that is an NGS readout that's RNA based can be paired with this. So if you use our custom panel that I think we have now up to 200 targets that you can order custom, you could have a very specific set as well as TCR with this set.
Excellent, and this really is a nice extension to GeoMx of the TCR profiling that we're able to do with the nCounter. So we have our TCR Diversity Panel with nCounter for human, and you can get a TCR diversity score on the bulk level. So you could, in theory, imagine a scenario where you would be analyzing hundreds of samples with the nCounter TCR profiling panel, and then the ones that give you really interesting diversity scores compared to the clinical information, you could go back and analyze, with GeoMx DSP, another serial section. So I wanted to get to you, Hargita. So can you tell us a little bit more about the samples that you tested out with this TCR profiling assay at Atreca?
Absolutely. So we decided to, at a high level, really assess the performance of the TCR spike-in panel against what we know of the Whole Transcriptome Atlas data. So we selected 2 commercially purchased colon specimens from 2 different patients, both stage 4 adenocarcinomas, and one of the specimens was a colon that we had run previously on WTA experiments, and the other colon number 2 was a colon that we decided to run on the TCR panel. It was new to our lab, and then we also ran tonsil as the control, and we selected 4 different regions of interest. The regions of interest we selected based on what we felt had a really clear signal of pan-CK tumor with a tumor microenvironment where maybe there was some immune infiltration going on. We further segmented the regions of interest into 2 areas of illumination. The first one was tumor, the other one was tumor microenvironment, so it was everything outside of pan-CK. And the regions of interest we collected were at the maximum collection size, which was 660 by 785 millimeters. And then we placed the collection onto our sequencer and gave the data over to Jason and the rest of the team on the TCR Alpha program to analyze.
That's a great segue into asking you about the analysis, Jason. So, how does the analysis work with the TCR profiling assay?
So, as Hargita mentioned, we're working very collaboratively as part of this Alpha program. And the Alpha program comes with a commitment that we're going to be supporting the analytical development that the customers who are participating in the program need. We want to learn about how the assay is performing, but we also want to support people in terms of interpreting the data on these biological samples that are so critical to them. So the way that we work with the data can actually happen in 2 different ways. Because this is a standard panel design for us, it works beautifully in the DSP-DA data analysis suite that is part of the machine that you get when you purchase a GeoMx machine. So you don't have to take it off the machine. Now, we don't sit in the same city, so we did need to actually transfer some of the data. And so you were so kind enough to provide me with some of the outputs to work with from the data analysis suite. And I analyzed some of the data with our open source bioconductor tools that's called GeoMx tools. That's a package that we released last year that allows you to do full end-to-end analysis, exactly replicating the type of work that you can do on the machine itself. And so the way that we look at this, when you're thinking about TCR, there's really 2 things that you really care a lot about. You're thinking about whether or not a clone is present, and then whether or not that clonality across all TCRs is expanding or contracting. You want to know whether or not the T cells are reacting to something. So we look for signal over the background probes that we have for understanding whether or not a TCR is present. Because you took very large ROIs, we have great selection of being able to see lots of different TCRs coming into different regions of the tissue. But one of the great things that we saw in your particular sample was that there was a handful of TCRs that looked like they're very tumor specific, and they only show up in the tumor niche that you selected based on your segmentation strategy.
Interesting. So what were some of the findings then?
So what we saw in that particular sample was there was a shift in T cell phenotype in terms of what T cells are present outside the tumor versus what T cells move physically into the tumor. The population that's outside the tumor was heavily CD4 positive and had a very specific fingerprint of the clones that were present. You'd see some of them coming in and out, up or down. But when you look at what actually migrated into the tumor, there was a set of T cells that were only CD8 positive now. All the CD4 was excluded. And you started to see a handful of variable and joining chain regions specifically show up that you saw once or twice in the stroma, but now became a whopping signal indicating that those are most likely the tumor reactive clones that are actually trying to stop the invasion of the tumor into the neighboring tissue.
Interesting, very interesting. So, quick follow-up question to that. Is it possible to potentially do this same analysis with this TCR profiling probe, say with the CosMx spatial molecular imager?
We are actively researching that. We definitely want to bring that forward to our platform. We're trying to make sure that we can balance the localization that CosMx gives you with the ability to interpret your data.
Uh-huh.
So if we bring this to CosMx, one of the great things you'll be able to see is the interactions between cells.
Yeah.
But one of the things that GeoMx does fantastically well right now is it gives you the whole transcriptome.
Yeah.
And so one of the things that we can look at with these T cells when we're using the whole transcriptome assay is what is its functional state.
Mm-hmm.
One of the things that we profiled after looking at the fact that there was an expanded clonotype was whether or not these T cells were dysfunctional. And we also saw
like potentially exhausted.
And so there's plenty of great exhaustion signatures that have been published in the literature, and we can take those and just directly use them with our GeoMx assay because we're covering the whole transcriptome. So CosMx is going to get you those really fine-grained interactions, and we're going to learn a lot about how those T cells that have invaded are physically interacting with their environment. And then we'll learn about their state with GeoMx because it gives you the full breadth of the transcriptome.
Excellent. So if someone who's watching is interested in, trying out the Tcr profiling assay through the Alpha program, so what should they do?
Well, obviously, feel free to reach out to us.
Mm-hmm.
We are recruiting more people to be participating in the Alpha program, and what we're trying to do in the very short term is release a couple toolkits that people can use when they bring their analysis on board.
Mm-hmm.
We'll provide them with reagents to run in their lab. So if you've got a GeoMx, this is something that could be available to you.
Mm-hmm.
And we'll provide you with the analytical toolkits to think about TCR based on some of the learnings that we have, both from internal and the lovely collaborations that we're doing with the folks like Arutha.
Excellent. And that's a great segue to you, Hargita, to close out. Do you think you're going to be doing any more experiments with TCR profiling?
Absolutely. This opens up a whole different world for us because we can actually start utilizing not just the PBMC samples that we have collected, but actually look at the tissue samples that we've collected for our clinical trial.
Mm-hmm.
So I think it's going to also increase the way that we look for novel targets in the future.
Excellent. Well, thank you so much, Hargita and Jason, for being here to speak with us today.
Thank you for having us.
Yeah. Hello, everyone. Elizabeth Schneider here, your NanoNews correspondent, coming to you live from AACR. I'm the Senior Global Marketing Manager at Nanostring, and we have a treat for you. I managed to grab our Chief Scientific Officer and Senior VP of R&D, Joe Beechem, right after his Spotlight Theater presentation. So Joe presented alongside our customers Arutha Kulasinghe from University of Queensland, as well as Hargita Kaplan from Tcr Inc. And Joe, as well as our customers, presented on both using the GeoMx Digital Spatial Profiler as well as the CosMx Spatial Molecular Imager to spatially profile tumor sections, to both look at RNA up to the whole transcriptome as well as protein at the tissue structure level as well as single cells. So Joe, for those of us watching who aren't familiar with spatial biology and with GeoMx DSP and CosMx SMI, could you quickly summarize what you presented in your meeting and how these two technologies work?
Yeah, thanks, Elizabeth. I think one of the key aspects of the GeoMx Digital Spatial Profiler and the CosMx Spatial Molecular Imager are the different distance scales that they probe by spatial biology. The GeoMx Digital Spatial Profiler specializes in functional whole transcriptome imaging of particular compartments inside the tumor in the tumor microenvironment. Whereas the CosMx Spatial Molecular Imager goes down to the single cell and subcellular level and sort of measures what I call the private life of proteins and RNA and how cells communicate with each other and how they self-organize. So the GeoMx is functional measurements at the whole transcriptional level of tissue compartments. The CosMx is the private life of individual cells and how they talk to each other. And both those spatial scales really are important because that's how you get tissue function. You actually need whole organized sets of cells work together to get a result. And then they organize through cell-cell communication. So those are how those two different spatial dimensions work. And that's why we built two platforms.
Excellent. And so for people who are interested in analyzing their spatial biology data, a lot of these data sets are going to be quite large. So is there an option or a plan for people to analyze the data sort of seamlessly and collaborate across the globe?
this is really an important thing. When you start collecting big spatial data, these are not data sets. You don't email these data sets or FTP them around. You actually need a place, a repository that allow you to share those kinds of data sets with the rest of the world, collaborate, and generate high quality publications on. So we've built a spatial biology portal where both of our platforms stream the data up to what's called a data lakehouse. This is a very generalized way to store high dimensional data of any type. And then we are in a cloud, and then we host that. And then all the image analysis tools you want to do, the scalable compute tools you want to do, and all your data is artificial intelligence and machine learning ready. So then when you do a big study and you want to collaborate, and your collaborators may be 6 different investigators from around the world, all you have to do is send them a little email with a link that will take them to the data lakehouse, and all those image analysis and collaboration tools will be there for them. So you never have to move it around, but at the same time, 6 people around the world will have the same detailed data and the analysis tools as you did, the person who collects it. So this really solves a huge problem in spatial biology, is how do I collaborate? How do I actually work on these publications? And this basically takes that whole problem and it just disappears. Now it's easy as an email.
Yeah, I mean, it's almost like we're at the same point in the next generation sequencing revolution, where people were generating these large data sets and trying to understand how to analyze them. And spatial biology, to me, is sort of the next frontier, or the next revolution in molecular biology. So if someone wants to analyze, say, COVID-19, like Arutha presented on some data that he had looked at in non-small cell lung cancer, but in additionally lung tissues that were from people who unfortunately passed away from COVID-19. If someone wants to work with, say, probes for SARS-CoV-2 or other markers, is there the ability to customize a GeoMx DSP experiment?
Yeah, this is great. So we actually left these barcodes open for just that purpose. So we do a whole transcription. You say, well, you got everything. Well, then sometimes you don't. You got an infectious disease. You want to make custom probes that look at SARS-CoV-2 or a whole variety of things. SIV was some of the data that we showed today on. So you can customize, make these probes. They get mixed into the whole transcriptional assay, and they all get simultaneously analyzed that way. So it's a very flexible way of putting unique content inside the whole transcriptome.
Excellent. And that brings me to my next question. So, Hargita actually presented on an assay that we have currently in sort of alpha mode, where you can look at TCR, T-cell receptor profiles spatially. So I was wondering if you could, for those who weren't able to attend the Spotlight Theater, if you could comment a little bit on that.
This is a super exciting area for us. So, it's a battleground. The tumor and the tumor microenvironment is a battleground. Either the immune cells are going to win or the tumor cells are going to win. And the immune cells that are like the stars of the show are immune cells that go in, usually CD4 and CD8 T cells that go in. And then when they have a cancer-specific target for that particular patient and they interact with that particular target, then what happens is that they'll get activated and they'll start multiplying inside that tumor. That's when you're going to win on the whole tumor versus the stromal sort of war that goes on. Now, there's a lot of T cells in there, but the ones that are going to save your life are those ones that what's called clonally expand. Mm-hmm. And historically, nobody had a way of actually looking spatially inside a tissue sample like that and finding the T cells that really matter, those ones that are clonally expanding. So we took our whole transcriptome assay. We designed probes specifically to measure T cell clonality. We put them in there, and now you get the whole transcriptome. And then as you go through the tissue spatially, you should be able to localize regions where you have T cell activation and an increase in clonality. And that was the data that Hargita presented today. Mm-hmm. It's really exciting, really never been done before spatially. And that's what we're spearheading with this new technology that we're adding to the whole transcriptome.
Excellent. So, protein is the other side of the coin. So I know GeoMx DSP was first launched with sort of 96 plex protein panels for nCounter readout, but with NGS readout, we can go beyond that. So what can the GeoMx DSP do in terms of number of plex for protein?
Yes, so it's nice. When we made these barcodes, we made them next-gen sequenceable on the antibodies. Now we have unlimited plex. The plex levels that we've been doing in the lab are roughly about 150. So you can have 150 antibodies that you're looking simultaneously, all spatially resolved. They all photocleave those barcodes. You go up to 150 plex. Then the other thing we showed at the meeting here today was that you can actually combine that with the whole transcriptional profiling of the same exact slide. So you can do 150 proteins and the whole transcriptome all on the same slide, spatially resolved now. So it's really kind of exciting development.
Wow, that is really, really cool. I think actually there was a poster presented on that yesterday.
That's right, there was, yep.
Yeah.
And if you're interested in that protocol, it's a white paper. You can go onto the Nanostring website and download it. See
Great.
exactly how to do that.
Great. So as we wrap up, I wanted to ask you about this initiative I've been hearing about at Nanostring called the Spatial Organ Atlas. So I know, in the days of single cell, when it was in its heyday, people were coming up with these cell atlases. So what is the Spatial Organ Atlas? Is it similar to that type of initiative?
Yeah, I think the one thing that hit us when we did the whole transcriptome assay for GeoMx was that there wasn't a database for these things. So if you look at tissues of the tissues of the body are all made out of these spatially resolvable functional units. If you're in a kidney, you got something called a glomerulus. If you're in a pancreas, things like islets of Langerhans, so on and so forth. And no one had ever done an in situ whole transcriptome of these functional elements of tissues. So when we were collecting this data, it's all brand new data. Nobody had ever measured these things. And I could look at the data and I could see all the discoveries we were making. It's like, we're not here to write 1 million papers, but the rest of the world may want to make 1 million papers out of that. So we took the whole transcriptome data, we put it in a public domain on the Spatial Organ Atlas. We actually also put a viewer around it. It's called the Minerva viewer. So somebody is interested in exploring a kidney or a lymph node or whatever can literally sit at home with a glass of wine and you can wander through and explore in all detail in an interactive manner whole transcriptomes all spatially resolved in these tissues. So it's really a great way to learn about how whole transcriptome profiling gets done in tissues. You learn a lot about tissue biology by doing that. And then it also serves as a reference database. So when you go and do your particular cancer, you may be interested in diabetic kidney disease, one of the cases we looked at, or you're interested in diabetes. You could have your particular samples, whole transcriptome them, and then reference back against these norms that are in the Spatial Organ Atlas. And you can immediately start looking for differences. Can I see early diabetic responses? Or can I see early effects of high pressure on your kidney? Mm-hmm. So those are all the things we're trying to enable with the Spatial Organ Atlas, and it's going to grow. We got about 6 or 7 organs in there now, and we intend to grow that out. We have a group in China that are actually doing liver right now, and I think we're going to be bringing that one on. I also presented here a whole developing mouse embryo that's going to go into the Spatial Organ Atlas also. So there's a lot of great data that's going to show up in the whole transcriptome data in the Spatial Organ Atlas, and I really recommend check it out. Look at the website and explore it.
Yeah, I mean, when you were mentioning, having a glass of wine and, exploring a kidney, it's really seeming to me like the Google Maps of different organs,
Absolutely. It actually looks that way. When you're actually looking at this data, you're actually hovering way above a tissue slice, and then you zoom in with your mouse, and then you're down at the single cell level. And I think the other thing is that there's a lot of discoveries to be had in that data. I already know people that are writing up papers that are just there. They didn't have to do any wet lab work at all. They just went right into our organ atlas and discovered what's in there and are writing it up. One of the things we discovered when we were first doing that, like just on the kidney, is that we discovered every transporter along the length of a kidney on our first experiment. They're all in there. You can just read them off. Wow. And there's a lot of things to discover in there. So there's a lot of stuff that's in the textbooks. There's a lot of stuff in this Spatial Organ Atlas that hasn't been discovered. The data's just sitting there waiting for someone to actually mine it.
Yeah, I mean, I think it's a great teaching tool, and it's also, as you said, a great way to understand what really healthy tissue looks like. I think we focus so much on the tissue in the disease state and what does the disease look like and how to treat the disease. But if you're really focusing on what healthy tissue looks like and what that early transition looks like, you can work more to actually prevent the disease in the first place, which is, I think, where we should be moving towards in terms of healthcare in general.
Yep. And that's where the whole transcriptome has an advantage, because when you actually look at that and you're looking at perturbations of that healthy state, you got 18,500 different things in a human, or 22,000 different things to actually look at in the mouse. And what you really do has this huge opportunity to see early biomarkers for dysfunction of normal tissue.
Mm-hmm.
And getting there early is what it's all about.
Yeah. So one final question, I was wondering if you could tell us a little bit about the sensitivity of the GeoMx assay. I mean, one of the unique features of the GeoMx DSP is that we're using these photocleavable oligonucleotide tags. And so we're not actually doing a poly A pull down. We're not actually sequencing the mRNA transcript. So, I believe that allows us to get sort of a wider dynamic range in terms of the levels of expression.
Yeah, the dynamic range on the system is really huge. It's like about 10 to 5.5 power, so it's almost 10 to the 6, so which is unheard of in a spatially resolved technology. And then the sensitivity level typically goes down to what they call the 1 FPKM level. So roughly about 1 transcript per cell, you can actually observe what the GeoMx whole transcriptional assay. And it's good at the low. One of the things we saw in the spatial organolis when we did the mouse is that every transcription factor, and they're all relatively lowly expressed. We observed every single one of them in the mouse organogenesis study we did. So you can go down there, get good signals out of your transcription factors, which are your low expressed, and then you can see all the other transcripts with it.
That's excellent. I mean, I think that really extends sort of the legacy of Nanostring, particularly with working on FFP, because we have the nCounter analysis system that also has a wide dynamic range, no cDNA conversion, no amplification, direct digital detection that does give you that wide dynamic range and the ability to detect really low expressing transcripts. So thank you so much, Joe. It was a pleasure as always. And I hope everyone watching had a chance to catch the Spotlight Theater. And if you are watching this live and you haven't had a chance to talk to someone at Nanostring and you're here in New Orleans, come on down to booth 1719 and chat with the experts in spatial biology.
Okay, good deal.
Thank
Great
you.
talking with you, Elizabeth. And
Yeah.
good luck at AACR. Love it.
Hello everyone, we're back for another NanoNews break at AACR in New Orleans, Louisiana for the afternoon poster session, day 3 of AACR. I'm Elizabeth Schneider, Senior Global Marketing Manager here at Nanostring and your correspondent for these NanoNews breaks from AACR. So today I'm with Sarah Church, Senior Scientist of R&D at Nanostring. She's going to be talking about a poster where we did a collaboration with the Mayo Clinic and OHSU to develop a custom high-plex GeoMx digital spatial profiler breast cancer protein biomarker assay. So Sarah, can you walk us through this? What was the impetus for creating this assay?
Yeah, so both the Mayo Clinic and OHSU were really interested in having a GeoMx-based assay where they can measure multiple proteins at the same time that are clinically relevant to breast cancer. So they developed a panel that is a custom 27 antibody panel. It covers all of the major breast cancer biology, all those major diagnostic markers such as HER2, all of the estrogen receptors, the progesterone receptors. And then there are a number of different other immune-related markers and proliferation-related markers. And that way you can do one assay on one piece of tissue and measure all of these different clinical relevant proteins together. So to sort of validate this assay, they ran it at 3 different sites, 2 different sites at the Mayo Clinic and then 1 at OHSU. And here is just looking at run-to-run variability for each of the different targets in the panel. You can see that they're very, very close together. So even with different instruments, different users run at different times, all of the assay was performing very, very well each run. And then the other portions are really related to FDA-approved biomarker assays for breast cancer. So the first one is looking at just at HER2. So HER2 IHC scoring goes from 0 to 2 to 3. So if you're HER2 positive, you'd have a 2 or 3 score. And so we just wanted to see that if you look at those scores based on IHC, how well that correlated to HER2 expression in this custom assay. And you can see that really significantly, as you go from 0 to 2 to 3, you have very clear and distinct expression of HER2 based on the DSP assay, showing that you can do what we do in the diagnostic assay also in the GeoMx assay. So that's really cool to see. The similar was done for both estrogen receptor and progesterone receptor. So the 2 different sites had 2 different clinical cohorts. One was ER positive, all ER positive and then HER2 negative, and then another one was both ER positive and negative and then HER2 positive. So here you just see that they're all ER positive. When you correlate that to the IHC ER, we have really, really high expression of ER based on our DSP assay. And then similarly in this cohort, the positive samples are much, much higher than those that are negative. And then very similarly in the progesterone receptor, you see that those that were positive have really high PR in the GeoMx assay compared to the negatives in both of those cohorts. So it's really nice to show that not only can you repeat this assay at different sites, but you can also really correlate the protein expression that comes off the custom assay with clinical assays.
And then remind me again, this was done with nCounter readout, right?
That's right.
So that's another advantage of having that reproducibility and reliability where you're not sequencing the tags, we're just hybridizing probes on the nCounter too.
Yeah, absolutely. You know, nCounter systems already FDA 510 approved, so that just reduces the variability in that readout. Yeah.
Excellent, excellent. And then can you walk us through how the regions were selected? What regions of the tissue were selected to profile?
Yeah, absolutely. So in the initial concordance assays, they basically selected the same ROIs at all 3 different sites. And they were really picked in order to have both coverage of tumor as well as stromal areas. So it's maybe a little hard to see here, but there's sort of pink areas, which are the CD45, and then the green areas, which are the tumor or the PNCK. And so they actually did segmentation based on the PNCK, so whether it's in the tumor, in the epithelium, or in the stromal areas. So not only did they look at each of the targets that are related to the tumor, but also related to the stroma and how those would be read out in a clinical assay.
That's really interesting. So I have another question for you. Like in the clinical version of this assay, I assume that they're looking at HER2 positive or negative based on IHC already, right?
Okay.
Or are there some instances where they use a bulk gene expression profiling assay to determine whether it's HER2 positive or negative?
Yeah, so the FISH assay is also one that's used to look at HER2 positivity. So these ones are, when we're looking at the correlation here, it's based on the IHC, but most of these were also FISH positive.
Okay, excellent. So what are the next steps for this work?
Yeah, so the next steps are, we have 27 different targets, so it's a much further analysis of all those targets and how they're relating to the different clinical pathology, and we're going to be writing this up and publishing it soon.
Excellent. So do the folks at OHSU and at the Mayo Clinic, would they like to use this for every breast cancer biopsy sample or for only certain ones?
I think, the data will say in the end, we have these 3 different cohorts that are really different breast cancer biology. And so, the things that relate really well are probably going to be more likely to be able to be used in some sort of clinical assay, as opposed to, some of them might not be as strong in the GeoMx. And I think there's also an extra value proposition, right? Because we're looking at all 27 of these targets at once. So, being able to look at different relationships between each of the proteins as well might be something that we eventually develop.
Excellent. Thank you so much, Sarah. It was a pleasure. I hope you enjoy the rest of the poster session. Thanks. Hello, everyone. Elizabeth Schneider here, your NanoNews correspondent coming to you live from AACR. I'm the Senior Global Marketing Manager at Nanostring, and today we have a special treat for you. We actually have one of our neighbors in the same building as the Nanostring headquarters in Seattle, Presage Biosciences. And this is Rich Klinghoffer. He's actually the CEO of Presage Biosciences. They're using the GeoMx Digital Spatial Profiler to do some really cool dosage studies with cancer immunotherapy drugs. So he's going to be talking today about his poster on mechanistic profiling of immuno-oncology drug combinations in cancer patients with the Chivo intratumoral microdosing and Nanostring GeoMx DSP. So Rich, tell us about this device. I mean, that's sort of the crux of Presage. So it microdoses multiple concentrations of the drug compound into a tumor?
So we built the CIVO technology to essentially build a better bridge between laboratory studies and clinical studies. So it's really to address a long-standing, I think, well-recognized issue that there's a discordance between results derived with investigational agents in the laboratory setting and then their performance when they hit the clinic. And we all know about this. 1 in 10 drugs makes it to FDA approval based on benefit. So we're trying to help out this situation. We're saying, look, if you want to develop drugs for humans, let's analyze those drugs and drug combinations at an early stage in intact human tumors while those tumors are still in the patient with access to all the biologies of that patient, and do it in a manner that's safe so that we can evaluate their biology early in development without harming the patient.
Excellent.
And so, do you want me to go?
Yeah, sure.
And so the way it works is it starts with a device that gets inserted into the tumor through the skin, and it delivers multiple columns, up to 8 different drugs or drug combinations directly into the intact tumor. All our patients are scheduled to have surgery as part of their care, so it's therapeutic removal of that tumor. Once that tumor comes out, we take that tumor and section crosswise to these injection columns, and that's where our analysis comes into play, and that's where we're starting to incorporate the Nanostring GeoMx DSP, which is a perfect complement to what we're doing. So now we can take all these trackable locations of our drug microinjections, and they're trackable based on a component of our technology, which are these little fluorescent microspheres, and create regions of interest for Nanostring GeoMx DSP analysis, and cast a wide net to see what type of responses are elicited to either single agents or combinations. And these can be investigational agents, FDA-approved agents, small molecules, biologics, or combinations thereof. So here, we're in this study. This is a study in patients with soft tissue sarcoma. We inserted a number of different agents, including the well-known checkpoint nivolumab, a classic version of a cytokine, IL-2, combinations of those 2 drugs, and then profiled using digital spatial profiler to look at responses. One of the interesting things we did is to start getting into better understanding of the complex interactions between the tumor cells and various cells within the microenvironment. Is in this case, we applied a T-cell mask to better understand what spatially transcriptional events are occurring in the T cells versus non-T cells. And I could tell you that based on enrichment of classic T cell marker CD3 itself, as well as many other markers of T cells, the mask using the DSP system worked really, really well. And here's just one example of a patient which received anti-PD-1, IL-2, and the combination of the 2. And we see several important events. We see in the combination, we see elevation of classic T-cell reactivation markers like granzyme B. But interesting, as you might expect it would happen in a tumor, you don't only see activation of pro-immune responses, you see feedback loops. And these include activation of immune suppressive markers like IDO1. And so it gives us an understanding of what are the events that we need to address with new therapies in order to overcome these immunosuppressive feedback loops. So here again, we see what we can capture, just a glimpse of what we can capture with DSP. And we validate that in our own labs with our own immunohistochemistry. Here we could see elevation of granzyme B in the T cell population. And then we could do other things. For instance, go back and look at new tumors. So this is a completely different patient. And again, similar to what we saw with DSP here, we could see elevation of pro-immune signals, like elevation of the chemokines CXCL9 and CXCL10 only in the region that saw the drug combination. But interestingly, again, we see the tumor trying to reach homeostasis by elevating the feedback loop, an immune suppressive event, IDO1. And you can see that when we quantify these, it's very synergistic when we look at the combination versus the single agents. Right now, we're using this as, again, a better translational oncology research tool. We do this in partnerships with various pharmas. The one that we presented posters on during AACR was our partnership with Takeda. We have 2 projects with them looking at their IO agents. And ultimately, we see a very intuitive utility of the technology as a functional precision diagnostic, or a diagnostic for drugs that have no clear genomic biomarker of response, so we can start teasing out exceptional responders versus non-responders. And we're already seeing evidence of this when we compare different patients using the signals that we get, or the signatures from the Nanostring platform. This patient number 18, a really nice responder, versus patient 21, shows a response in the immunosuppressive direction, but not the immune pro-activation direction. Yeah, and we think ultimately we may be able to use these signals to direct a cancer patient's care with specific drugs. So I hope that gives you a nice summary of what we're trying to do with this technology.
Yeah, it's really cool. It's almost like you can do 8 different clinical trials.
That's exactly right. You can do, so all your arms of your different trials, again, up to 8, can all be done in a cancer patient's tumor. And then we do it across a number of different cancer patients so that we can better understand, like I said, patient-specific differences that may ultimately not only highlight why some patients respond better to others, but also potentially direct their care in the future.
Now, a quick question for you. In this example, you said you would inject it through the skin.
Correct.
So that would be a case where the tumor is like subcutaneous.
Correct.
It's in the soft tissue. But if it's something that's like more in the internal organs, would the surgeon do this before they actually remove the tumor and do the injection then?
They would. And so, now that we have established the technology with our 1st generation device, of course, we're working on new iterations, 2nd generation devices. Our next generation, which we call our deep reach device, where all the needles are encased in a 16 gauge introducer, is now in canine studies.
Oh, excellent, excellent. Nanostring, as you may or may not be aware, actually has a canine immuno-oncology nCounter panel.
Oh,
yeah,
that's fantastic.
What's really interesting is for the viewers who are watching this live, a lot of canine oncology patients are not actually, lab animals. They're actually real patients.
Absolutely.
People's pets that develop spontaneous cancer. And canine is a great model for human cancers because they develop a lot of the same cancers spontaneously. So that's really cool.
As a matter of fact, when we moved the technology from the laboratory settings, so just mouse models of cancer, into the human clinic, we did segue through the dog clinic. We tested the technology out in 70 dogs with cancers, mostly soft tissue sarcomas and lymphomas.
Well, I'm sure all the families of those dogs are very happy if they were able to really pinpoint what drug combinations worked best for those patients and those dogs.
Absolutely.
So I was wondering if you could tell me a little bit about the assay that you used for the GeoMx. Did you do, obviously you're doing RNA, did you use the whole transcriptome atlas, did you use something else?
For this study, we used the Cancer Transcriptome Atlas. That's been our workhorse thus far. We have started using Whole Transcriptome Atlas as well, but mainly we use Cancer Transcriptome Atlas because, again, that's what we
has all the pathways.
Has all our pathways, is our workhorse, and we really like the fact there that you have a nice probe set with multiple probes for different genes.
Yeah. So my next question is, everyone is trying to move towards organoid development, where you would take like a biopsy of a tumor, grow it in a lab in 3D. Are you guys looking to do some experiments like that?
So we're not, and I'll tell you a couple reasons why. One reason, lots of other groups are doing it, so you really don't need Presage doing that. 2, we're trying to really emphasize the importance of evaluating drugs in a minimally invasive way, in a minimally manipulated way. So Look, I really appreciate all the work that's going on with organoids, but we want to know what happens directly in that tumor in its native state while it's still in the patient without any further manipulation than a needle stick and a drug microdose.
Yeah, no, I definitely see that. But could you see, like, perhaps in like a research lab, someone might want to use your device in that manner?
Absolutely. So in the research lab setting, you can still generate really important information, particularly using DSP. And I'd say there that you probably want to use more advanced mouse models that are immune competent so that you still can capture the complex interactions that occur between multiple different components of the immune microenvironment once a drug hits that environment, and compare drug exposed versus unexposed. And again, I think that's the beautiful application of combining what we do with the Nanostring technology. I mean, it's layering drug exposure on top of spatial biology and saying what happens when you have a drug combination versus a control region versus a single agent, and making those comparisons in a very spatially defined way.
Yeah. So I wanted to comment a little bit on your region of interest selection. So it looks like you did geometric regions of interest,
Yep.
but I know that we could also do sort of like contour profiling, so kind of like a bullseye pattern.
Yep.
Is that something that you've done in other
We
studies?
have, and so, and it's actually quite striking. So the nice thing about what we do is when we insert the drug does what you would think it would do. It diffuses around that epicenter of injection. What that creates is a gradient of drug from high to low. So in a sense, every site of drug microinjection can result in its own concentration response curve. So you don't need to look at different concentrations at different sites. We have set up our ROIs for DSP like that, and indeed, we absolutely get
A dose-dependent response.
a dose-dependent response with R-squares that are quite unbelievable in some cases. I mean, 0.7 to 0.9, and so it's very, very accurate.
That's amazing. This is so great. I mean, like you said earlier, it's almost like the GeoMx DSP was tailor-made for your application.
I humbly say I think it's the best application of DSP.
Yeah, excellent. Well, thank you so much, Rich. It was a pleasure. And I definitely hope to run into you in our building
Absolutely.
in Seattle. And thank you for explaining your poster, and I hope you have a good rest of the poster session.
Thank you so much.